It is my pleasure to introduce today's speaker, Dr. Pablo Milla Carmona. Pablo completed his undergraduate degree at the University of Buenos Aires, where he also went on to earn his PhD in geology. Following this, he joined the University of Bristol as a postdoctoral researcher with the Bristol Paleobiology Group before returning to Argentina, where he is now based at the Instituto de Biología de Organismos Marinos in Puerto Madryn. Pablo's research lies at the intersection of paleontology, evolutionary biology, and quantitative morphology. His work focuses on understanding the processes that drive morphological evolution. He combines geometric morphometrics, theoretical morphospaces, and computational methods to investigate how form, function, and ecology have shaped evolutionary diversification. Pablo has also made significant contributions to computational paleontology by developing tools that help researchers analyze morphometric data. So most recently, he helped to lead the development of the R package morphospace, which provides a comprehensive workflow for constructing, visualizing, and interpreting morphospaces. Today, Pablo will be introducing us to this R package and demonstrating how it can be used to investigate evolutionary questions in his talk titled Investigating Evolution Using Theoretical Morphospaces, a Practical Workflow with Morphospace. So please join me in welcoming Dr. Pablo Milla Carmona to the stage. Take it away.
Thank you, Erin, for the introduction. Indeed, I'm going to be talking about morphospace, the R package. But before that, I'm going to talk a little bit about theoretical morphospaces. The theoretical morphospace approach was first proposed by David Raup during the 60s. It was a framework for the study of morphological evolution that was based in this really cool method he devised to quantify the geometry of the mollusk shell as variations of the logarithmic spiral. Raup then explained how you could use this model to by varying the parameters of the logarithmic spiral, simulate or produce the full range of possible shell morphologies, right? Including some that, even if are possible in principle, you wouldn't be able to find in nature because they either went extinct or never evolved. This is what we understand to be a theoretical morphology. And the end game of this framework was to use these parameters to build a mathematical space that would contain the full spectrum of possible geometries. This is what we call a theoretical morphospace, which we would then be able to use to assess the distribution of empirical morphologies together with and in the wider context of all the morphologies that were possible. Now, that was not something you would be able to do with the morphometrics of the time, but now we have a geometric morphometrics. This is a family of methods that includes most notably landmark analysis within the Procrustes paradigm, as well as others like elliptic Fourier analysis, methods that have in common that they are rooted in a fully operational definition of shape, understood as everything that is left after we remove the effects of position, orientation, and scale. And well, these methods have shape and size analytically separated. But crucially for this talk, these methods, they all preserve the geometric information throughout the analysis. This means that at any point of your geometric morphometrics pipeline, you should be able to take the numbers you're working with and translate them back into the morphologies they represent. And I think it's fair to say these methods became the standard for testing hypotheses about the processes that affect morphology. But an aspect that I find to be a little bit underappreciated sometimes about geometric morphometrics is that it provides a way to simulate theoretical morphologies. This is because even if geometric morphometrics analysis are built upon empirical data, the method still grants you access to a mathematical space that contains all the possible geometric configurations that you can achieve with a particular account of morphology. So this is actually a very powerful and convenient way to implement the theoretical morphospace approach of Raup. And I think we can see this in how common theoretical morphologies are in the geometric morphometrics literature, right? We see them in these shapes we use to represent your shape change along PC axis. And whenever you see a mean shape calculated for a group of specimens. We see them in the phylogenetic nodes of a phylomorphospace, and of course, in the main application of this kind of thing, which is the adaptive landscape, in which we are able to map performance onto the morphospace because we have measured that performance on theoretical morphologies covering the entire space. So you can see that this is something people have been doing for a while, but for, I think, historical reasons, I'm not going to go into in this talk. Very few of them would say they are working with theoretical morphologies or theoretical morphospaces at all. And since my own research was more and more about working with theoretical aspects of shape variation, because I was interested in the relationship between intraspecific and interspecific variation, I ended up writing morphospace and R package in which I integrated all these tools into a workflow that I will now explain. But before going there, I just want to clarify what the package does and what it doesn't. So morphospace is only concerned with the stages of the geometric morphometrics pipeline of shape data, ordination, and visualization. It does nothing on the fronts of digitization, normalization, or the many statistical tests that exist for shape data. For those, you should use packages such as Geomorph, Morpho, Momox, and others, which, by the way, Morphospace is built to work together with. So going to the workflow itself, the first step of this workflow is to clean your shape data so you can remove some sources of variation that are embedded in it that can be acting as noise. In the following, I'm going to be using this layout. You will have pseudocode at your left, which is intended for you to follow, the procedures and indicate some relevant functions and their arguments. And then you will have the intended graphical output of that pseudocode on your right. And the starting point of this workflow is a set of shapes that you must have obtained using other means. So, for example, this is a 3D snake skull data set, which was micro-CT scanned, and then 3D landmarks were digitized using MeshLab before being imported into R, where they were superimposed using the proxim function from Morpho. But you could have easily used, say, GPHM from Geomorph, whatever gets you your shapes. And I'm using a couple of morphospace functions here, but they are not relevant at this point. What I want to show you is how shape data look when it's entering the analysis, both in its shape format, right? We have the superimposed landmarks and the wireframes connecting them, as well as the shapes as data points in a morphospace. And shape data at this point can be quite noisy because the variation that you are observing is produced by several different processes, some of which can be, masking or even distorting some of the signals that you are interested in studying. And so, for example, in the case of the skull of snakes, my understanding is the bones of the skull are quite mobile, so asymmetry can become something of an issue. So it's really convenient, for example, that Geomorph has this function by that symmetry, which if we indicate which landmarks go into which side, will separate the symmetric and asymmetric components, so we can keep the first and discard the rest, right? So you can see that this is the original shape variation. You can see it looks, well, more symmetrical, of course, and cleaner, and the variation also shrinks because you are getting rid of variation that you're deeming unimportant. There are other biological processes that can be acting as noise. For example, we all know that ontogeny can introduce substantial shape differences, but unless you are specifically interested in studying ontogeny, those differences can be masking, for example, more subtle patterns that you might be interested covering. So with that in mind, we have the different shapes function, which allows you to remove any sorts of variation that you can model using a linear model, right? So that's those are regressions, ANOVAs, and the like. And that's what I'm doing here. I'm using the LM function to regress shape on size, and then I'm using the trend shapes to remove ontogenetic allometry. And again, you can see the shapes look considerably cleaner, and the variation shrinks a lot. So you can see you can use a number of different functions from different packages to remove a noise from the shape data before you submit it to the second step of the workflow, which is the ordination of this shape data and the representation of shape variation. Now, I mentioned earlier that shape data already exists in a mathematical space that contains all the possible geometric configurations, right? That is a shape space. But the catch is these shape space are usually high dimensional and very difficult to read, at least for a human. So what we would do in geometric morphometrics usually is to apply some form of ordination analysis, which will summarize shape variations such that you can represent a meaningful portion of it in a low dimension representation. And by far the most commonly used method for that is principal component analysis. And this is how this step looks in the morphospace pipeline. We have mspace, which is the core function of the package. And what this function does is you feed it with the output from another function that performs ordination, right? So for example, here I'm using the prcomp function from stats to PCA to perform PCA on the symmetrized shape variation. And then I'm feeding mspace with this object along with other information about the shape dataset. Like these are landmark data. We have 71 landmarks in 3 dimensions, etc. And what mspace will do is to produce this bivariate plot, which is empty in the sense that it has no data points or anything like that. But you see shape variation mapped onto this space using these theoretical shapes in the background. This is a rather common strategy in geometric morphometrics, right? So these theoretical shapes are indicating us how shapes in different regions of this morphospace look, right? And at this point, you can represent shapes using different, I like to call them accessories. In this case, just a wireframe connecting the landmarks, but we have other alternatives. For example, you can use a 3D mesh, which is then warped onto the different configurations. It's the same variation, but it's represented, I hope, in a way that is easier to read. And once we have all our, once we have produce these ordination axes that have a shape variation mapped onto them. This, remember, is an empty morphospace. We're going to start projecting other stuff in there using another set of functions, which I'm going to explain in a minute. But what I want to show you right now is this PCA projection with these 3 groups of shapes, which in this case represent clades. This is groups of a species, but it could be any other meaningful categorization of the data, like, I don't know, stratigraphic levels, ontogenetic phases, ecological categories, whatever, right? And say we are interested in assessing the differences between groups in terms of shape. And if that's our aim, this projection is not really useful. This is a projection which is, since it's made using PCA, is maximizing total variation, right? But this is not very useful here. We cancel a lot from this particular projection. Luckily for us, there are other ordination methods which will maximize criteria other than just total variation. For example, we have the between groups principal component analysis variant, which will maximize a variation or separation rather between these groups that we know beforehand. I'm performing between groups PCA using this BGPRComp function, but which is morphospaces, but you have others, for example, alternatives in Morpho. Those are all compatible with the morphospace package. And what I'm doing, I'm using just the same shapes, and I am providing a classificator. And other than that, I'm just projecting the same shapes and the same groups, right? But you can see how big of a difference this projection makes in terms of interpreting the differences between groups. We now see much more clearly the separation between them, and we can assess the differences between them among them in terms of shape differences, which are much more clear now. And we have other such methods, partial least squares, phylogenetic PCA, and others, which when you use them, what you're effectively doing is rotating the shape space to a viewpoint that facilitates reading some certain, signal, biological signal that you are interested in studying. So if we go and just do a PCA of these data points, this is perceptual art, but I like the metaphor. We would probably see this angle, right? Because it's only maximizing total variation. But that doesn't mean that we don't have a clear signal in another, rotation of the morphospace. It's just that that signal is not aligned with the axis of maximum variation, right? So in our example before, we have clear separation between groups. It's just that these differences between groups are not aligned with a regular PC axis. We need to use a different rotation. And so after we have cleaned our shape data and we have chosen a useful rotation for the shape space, we can go to the final step of this workflow, which is to populate this empty morphospace by projecting different elements into it. These elements are points, lines, polygons, surfaces, and other objects that by virtue of existing in a theoretical morphospace are made of actual shapes, right? And we can use these elements to characterize shape variation, whether that is theoretical or empirical. We can use it to highlight some aspect of the distribution of shapes. We can use it to condense or emphasize certain patterns that we are targeting. They have many applications, and it will depend on your research question. So in the morphospace pipeline, this is done by appending one or more of these different Broch functions to the mspace function using the pipe operator from McGrader. And the idea is that these functions with a logic of layers, right? So each layer will represent a different element that has been projected into the morphospace. We have already seen a couple of these in action. The simplest of them is Broch shapes. This is a function that will project any number of individual shapes as data points. It doesn't matter if those are empirical or theoretical, as long as they are compatible with the morphospace, meaning they need to be measured using the same variables. In this case, for example, the same number of landmarks, and ideally superimposed in the same instance and stuff like that, right? Another function that we already saw is Broch groups. This, again, what you need to provide is a set of shapes and a classificator for them. And this function will project convex holes or confidence ellipses, delimiting the position of different groups of shapes. Then we have more complex elements that we can project. For example, Broch phylogeny, as you can probably imagine, will produce a phylomorphospace. Again, you need to provide a set of shapes that will function as phylogenetic terminals, and we also need to provide a tree that will be relating those terminals, right? Another function that I use quite a lot is Broch axis. you can see how you can start combining these functions. Projaxis, what this function will do is to project a linear trajectory in this morphospace, linear trajectory that is captured by, or rather as the output of either a linear model or an ordination method that has been performed on shape data. So for example, here what I'm doing is regressing a shape on size for each one of these groups separately, and then I'm projecting these trajectories using Projaxis in three different separate instances. And you can see, for example, that some trajectories are parallel, some are orthogonal, and we can use this tool to represent phenomena like allometric trajectories, which is the case here, but also stuff like, say, evolutionary lines of least resistance, which we are going to see in the demonstration with a little bit of luck. And well, the final approach function I want to talk about is approach landscape. These obviously produce a performance landscape, a surface that you can use for studying, say, adaptive evolution. This one has a little bit of a different mechanics to it, because you need to first produce the morphospace, extract the shapes, calculate performance, and then project them again. We are going to see that in the demonstration in a minute, but yeah, this is another tool that you can use to for example, study adaptation, and that is broad strokes the morphospace workflow. This workflow is intended to help you, generate nice figures for your publications, but hopefully also help you explore your shape data and help you, devise your hypothesis. And this is not to say that you shouldn't go and run all the statistical tests that you can. I always look for statistical backing for the patterns we are seeing, but hopefully this can function as a heuristic tool for you to explore your data. There are other aspects, stuff that I included, but I'm going to just go very quickly about them. You can use this plot mspace function to regenerate the shape space, the morphospace, or you can use it to also to combine shape variables with other kinds of information. For example, you can combine it with a phylogenetic tree to create this phenogram. You can combine it with categorical variables to create these box plots or violin plots, or you can combine it with other numerical variables to produce an hybrid shape size morphospace, for example, in this case. And that's it with the presentation. I'm going to go now straight to the demonstration. If nobody opposes, okay. Apparently not. So for the demonstration, I'm going to go into R in a minute. But for the demonstration, I'm going to use the Tyrannosaurus dataset, which is not it's not a fossil data. I apologize for that, but this is a dataset I use a lot because it's nice to showcase the capabilities of the package. Tyrannos is a genus of passerine birds that has 13 species that you can find in North and South America. And they most of these species have what you would call like a regular bird tail, right? Something like what you're seeing here in the center. But we also have these two species of flycatchers which have evolved these very extremely elongated tails independently, so a clear case of convergence. And since we know that the tail of the birds is shaped by both natural selection acting on aerodynamic performance as well as sexual selection, this is an interesting study case for exploring different processes. In terms of the data itself, we collected 9 two-dimensional landmarks on the tails of 281 specimens belonging to all the 13 species of the genus. So we had both intraspecific and interspecific variation. And we also had the phylogenetic relationships, which we took from the literature, as well as some other accompanying information. Okay, so going now into R to show you a little bit of code, and not only pseudocode, as I've been showing so far. I'm going to start by loading the dataset that I prepared beforehand, and then the functions that we are going to be using in this session, right? So morphospace, geomorph and morpho, the same packages that we used in the presentation, as well as phytools for doing all the phylogenetic operations. So this here is the data. We have the TPS object, which is storing the results from digitizing landmarks in the TPS dig software, right? And the TPS file was just imported into R using geomorph functions. This is how this looks like. We have one matrix of 9 rows for each one of our specimens, 2 columns, right? And as many matrices as specimens. So if we ask how many dimensions this object has, we can see it has 9 rows, one for each landmark, 2 columns, 1 for each dimension, X and Y, and then 281 slides or specimens. We also have this links object, which is just a list indicating how you should connect the landmarks in what order so you can more easily see details. So for example, here we can use by shapes to see how our raw data looks. This is not shapes, right? This is just the way these landmarks were digitized, and you can see it's a mess. But the wireframe, the links help a lot. We also have the phylogeny, right? Which we can visualize here. And we have some classificators for the specimens. So the classification into different species for each one of our specimens, as well as a classification for the sex of each specimen, so whether they are males or females. And well, that is the dataset. And now I'm going to start with the workflow. As I said during the presentation, we need first to get our shapes. So that's what I'm going to do using the proxim function from Morpho. And then I'm going to extract the shapes from this object, which has lots of stuff in there. And what we need is here, the ORP data slot. And so if we go and compile these shapes, we'll see that they now look far better, right? So these are actual shapes. And we also will extract the sizes, right? That we will also calculate the logarithm of them, right? So we already did this. And I'm going to use going straight to use the mspace function to show you how the morphospace looks like. Right, so this is the morphospace that we create with the mspace function. You won't be able to see it so clearly unless I rotate the background models, but you can see now that these tails, the variation that is being captured is a little bit asymmetric. This is because the feathers were not fixed, so some asymmetry pervaded the data. So I'm going to use, oh, by the way, this is another way in which you can use the mspace function, just providing your shapes directly instead of an object that is the output of a ordination method. But this will only perform PCA, right? But it's all we need at this point. So what I'm going to do is to use the bilat symmetry function from geomorph. provide an index for each one of our specimens. Apart from the shapes, yes, the object has symmetry and the landmark pairs right and left. These are just objects I prepared beforehand, which tell us which landmark belong to which side, right? So I'm going to run this analysis, and then I'm going to extract the symmetric shapes from this object. We have both the symmetric and asymmetric components, but we only want the first one, right? So I'm going to switch now to using the other way, the other mode of for using MSpace, which is by filling it with the output of the PRComp function in this case. Here you have something I didn't show in the pseudocode, which is if you want to make a PCA out of shape data, you need to put it into this, I like to call it long matrix format, right? So instead of having one matrix for each one of your specimens, you will have now your specimens, each one in one row. So the first two values are going to be X and Y values of the first landmark. The third and fourth are X and Y values of the second landmarks, and so on. So I'm going to do this, specify the kind of data we are using, which is landmarks. We have landmarks in 2 dimensions, 9 landmarks, no links, and I'm also going to rotate the models. And then project the shapes, the symmetrized ones, and the classificator for species to mark the groups, right? Just to show off a little bit. And this is how, well, this is how this looks like. You can see that asymmetry has been largely removed. And we can make this this morphospace a little bit prettier by adding colors, for example, 2 points. Right, so we can also add colors to the groups. We can change, we can switch from convex holes for the groups to ellipses, right? So you can do lots of fun stuff, I hope. But since we also have the phylogeny, we can produce a phylomorphospace. For doing that, we need to calculate the mean shape of each one of these species, which we will do with this expected shapes function, right? So I'm going to store it in this subject, and then I need to provide the symmetrized shapes and the classificator, and the expected shapes will, then calculate the mean shape for each one of our categories in this classificator. So we now have 13 shapes, one for each species. And I'm just gonna copy this because I want to reproduce the same morphospace, but here I'm going to include the species shapes and the phylogenetic tree, right? So this is a phylomorphospace. I hope not difficult to get here. However, despite the fact that we have our phylogeny projected into this space, it's still just a PCA projection, right? So it's just maximizing variation with no regard to the classification or the specimens or the phylogeny. So since we have a phylogeny, we could do something a little bit more interesting maybe, and that is to keep just the non-phylogenetic component of shape variation. This is achieved using phylogenetic PCA, which is a method that will, calculate the component of shape variation that is independent of a phylogenetic covariation or a phylogenetic signal, if you want. And that can be quite useful in the context of an adaptive evolution study, because we can get rid of the similarities and differences that are related to common ancestry, and explore similarities and differences between morphologies that are devoid of evolutionary or historic variation. So I'm just going to reproduce this morphospace here. I'm going to get rid of this part. I just want to find a morphospace. There you have it. And then I'm going to go and perform phylogenetic PCA using the fill PCA package function of PhyTools, right? I'm gonna call this PCA, the phylogenetic tree. Notice how, again, we need to put the shapes in the long matrix format using the geomorph function to the array. And then, again, I'm just copying this. But I'm gonna change the ordination object, right? Yeah. We get some warnings, don't worry about them. And this is the non-phylogenetic component of shape operation in these two first principal component axis, phylogenetic principal component axis, actually. And you can see that it's a little bit different to the other one. Actually, it's like the y-axis is inverted, so I'm going to invert the y-axis here, and this makes easier to compare the two ordinations. And we can see that the phylogenetic PCA ordination is a little bit different, not a lot, but enough so the signal can change. And now we are going to try and attempt to create a surface out of performance values, which is the mechanics I was talking to you earlier. And to do that, we are going to extract now the different theoretical shapes that we see in the background. We can do that with extract shapes, right? So I'm saving these shapes here in this shapes theo object. And then I just need to provide an MSpace object, which we can create by saving this morphospace into a separate object, right? So it will tell you information about how this morphospace was created, but we only want it so we can extract the theoretical shapes in the back. Right, so this object now has 20 matrices because we have 20 theoretical shapes in the background. And now I'm going to proceed to calculate the functional values, right? So I wrote this compute LD function, which will calculate the lift to drag ratio, which is a way of measuring aerodynamic efficiency for the tails of these animals, right? So I'm going to apply that function, but I think a more common way in which you will end up using this function is you extract the theoretical shapes, you export them, and analyze them in a different software that can do I don't know, FEA or CFD or other biomechanical analysis, right? So but here I'm going to go and calculate the performance values within R. So I'm going to take, again, these shapes as in long matrix format. And the margin one means whatever the function I put here will be calculated over each row of this long matrix. And the function will be repeatedly, right? And I'm going to save the values in an object. So here we have our 20 functional values. And now I'm just going to reproduce the same morphospace and add the surface. It is very important that you always keep your functional values together with the shapes they were computed from, because this will just project the surface. But if if you change the order of the shapes or you change the shapes at all, then the functional values that you calculated will not correspond to the actual shape, so that can create some problems. But here you have a surface that we almost cannot see. So I'm going to change the palette. A little bit better. We can actually use other displaying options. Right, this produces field contours. We can also, something I do a lot, I should include that as a default option in these cases, is to use both of them, the contour and the field contour surfaces, so it's easier to read. There, a little bit, maybe we can make the lines a little bit, easier to see. So this is the surface that we calculated. For you to make it to cover more of the morphospace, you should go to this part and probably change the limits of the morphospace. So the theoretical shapes that covered it cover more of the area of this axis. But for the purposes of this presentation, it will serve. You can see that most of the species with the regular tails are close to this adaptive optimum, right? So an optimum of aerodynamic performance, and that makes sense because we know that that species with regular tails are probably under a process of, natural selection on their capacity as flight devices, the tail, I mean. And so this makes sense that these species are sitting on this adaptive optimum. It also makes sense that our fly catcher species are not great flyers, at least not their tails, which makes sense because we know that these species are under processes of sexual selection, which can mess up a little bit biomechanical performance. But we learned something that we maybe didn't know before, and that is that there's one particular species that is much better. It has much better performance than the other. So if we wanted to know which one of those species we are talking about, we could use roach shapes. And I'm gonna enable like labels, right? So we see that the species Terran of Savannah is it has higher performance than the tail of Tyrannosaurus. Well, I think we are already running out of time, but just to show you a little bit of what you can do with these different rotations, I'm going to very quickly, I hope it makes sense, example in which I'm gonna first subset the data. I'm gonna exclude the flycatcher species because they are so different to the rest of the species that they are probably biasing the ordination a little bit. So I will keep only species with normal tails. And then I'm going to do a phylogenetically aligned component analysis with the GMPR comp of geomorph, using only the subsetted data set. This is the phylogenetic tree with 11 species and the shapes of those 11 species. And then I'm gonna create the morphospace with this PACA object, which was calculated using only the species with regular tails, but I'm going to project all of the 13 species. And you can see that these morphospaces is very different, right? It's showing us something different. This is the phylogenetic component of shape variation. This is the complement of what phylogenetic PCA does, right? So it will preserve the component of shape variation that is maximally covariant with a phylogenetic signal. And this can be useful, for example, if you're interested in exploring, the structure of shape variation beyond their functional aspects, right? And very quickly, I'm going to use the intraspecific variation that we have to calculate a couple of axes for the flycatcher species. An allometric axis, this is static allometry, not ontogenetic allometry, because our specimens are all adults, as well as a sexual dimorphism axis for each one of them. And I'm going to now project this axis into this phylogenetically aligned morphospace together with the first component of PACA itself, which we interpret to be an evolutionary line of least resistance for the cluster of species with irregular tails, right? So a direction of evolution that is preferentially followed each time that a species splits into two, and that accumulates over time. And we also have, the interspecific axis of variation of these species that we know to be evolving under a process of sexual selection. And we can see that these species, beyond the fact that they are, convergent morphologies, it looks like the process of sexual selection that is going on, it's amplifying divergence that was already present in the group of species that have regular tails. So you can use these tools to explore different aspects of your shape data. I hope all of this makes sense and that is useful somehow. And that will be it.